GC-MAF. Claims about GC-MAF have been made all over the internet. GC-MAF is a substance that has been touted as a magical cure for everything from HIV and cancer to autism. Some people claim it has an 80% success rate in treating stage 4 cancer, but the scientific community has rightly been skeptical about such claims, and scientific papers have been redacted due to unsubstantiated claims. This is the unbiased story of GC-MAF. It would be easy to dismiss Dr. Nobuto Yamamoto as a snake oil salesman. The claims in his papers have been called out and unsubstantiated, but looking into his scientific career, you find an accomplished, award-winning scientist and immunologist. Dr. Yamamoto isn't someone you think would put his career on the line for junk science. His first paper was published in 1957, and since then, he has been published in some of the world's most prestigious journals. In 1964, he was published in the journal Science and in Proceedings in the National Academy of Sciences. Dr. Yamamoto holds a PhD in biochemistry from Gifu University in Japan, has been a US-based researcher since 1959 when he was invited to be a visiting associate researcher at the Fox Chase Cancer Center in Philadelphia. He was later hired as a researcher for the National Institute of Health in Maryland and worked with Temple University as a professor. In 1987, as a professor at Hahnemann Teaching Hospital, Dr. Yamamoto published a paper in the journal Cancer Research that would become the basis for the concept he later termed GC-MAF. In 1993, Dr. Yamamoto officially left academia and took a position at the Einstein Medical Center in Philadelphia, where he still continued to research and write about GC-MAF. Dr. Yamamoto left Einstein Medical Center in bad standing over GC-MAF. Details of the breakup with the Einstein Center aren't readily available, but it seems to have to do with claims made over GC-MAF. In 2006, Dr. Yamamoto started publishing multiple papers on GC-MAF and Nagalase. The most notable of these papers, published in the International Journal of Cancer, claimed metastasizing breast cancer cells had been successfully destroyed with GC-MAF. Another 2008 paper, this one published in Cancer Immunology and Immunotherapy, claimed that GC-MAF was effective as a treatment for colorectal cancer. A 2009 paper published in the Journal of Medical Virology made the astounding claim that HIV-infected patients were rapidly cured by GC-MAF therapy. By 2015, these papers had all been redacted by the journals that published them. The Anti-Cancer Fund had been asked to look into GC-MAF and found that the researchers did not actually measure tumor growth or decline directly, but instead relied on Nagalase, which the researchers acclaimed as an accurate measurement of tumor load in a human body. This claim, while interesting, is far from proven and accepted. Furthermore, the studies lacked an institutional review board and medical records to back their claims of regression. Then, a company called Immunobiotech, ran by a man called David Noakes, made claims of an 80% plus cure of stage 4 cancer. Noakes was arrested and eventually put in prison, but the combination of claims made by Noakes and the questionable research of an esteemed scientist nearly made GC-MAF disappear. But what if there really is something to GC-MAF? What if Yamamoto was onto something, but used a bad method to try and prove it? What if disease cells really do express Nagalase and it keeps the immune system from allowing macrophage activation? Many times when science or treatments are discredited, the entire idea is thrown out. But there have been many papers published by other scientists outside of Dr. Yamamoto. Frame Biotech was approved by the FDA for a phase 1 trial, meaning that it had undergone extensive preclinical studies showing the mechanism of action and the ability to kill cancer. There have been over 60 peer-reviewed studies that have looked into GC-MAF and Nagalase. Surprisingly, none of the studies have been able to debunk the activation of macrophages by the vitamin D binding protein during the inhibition of Nagalase. It has also shown that Nagalase is raised in patients with different diseases from HIV to cancer. Could the nagalase enzyme keep the immune system from activating against cancer and disease cells? In 2020, Japan has multiple clinics set up with the injectable GC-MAF, and all versions are available online. Chipsa Hospital would like to further the study of GC-MAF and better identify the mechanisms of action. Chipsa Hospital has partnered with Legacy Labs, a Mexican laboratory company, and leading cellular scientists to make what we call cellular nagalase inhibiting factor, known as CNIF. The initial research shows that nagalase is an extracellular matrix degradation enzyme that is secreted by unhealthy cells and cancer cells during tumor formation and growth. This enzyme does in fact inhibit vitamin D binding protein macrophage activation. Legacy Labs has a patent-pending process that reduces nagalase through the expression of the GC protein from select cellular lines. By expanding and multiplying the expression from the cell, we can easily replicate the multiplication. We've witnessed the cellular nagalase inhibiting factor initiates programmed cell death and secretes pro-inflammatory cytokines produced by our own immune system, including tumor necrosis factor. Chipsa is also studying the combination of cellular nagalase inhibiting factor with VG5000, due to VG5000 being an anti-geogenic treatment and CNIF also showing these properties. Activation of macrophages and other aspects of the immune system, along with the targeted aspects of VG5000, show great promise. Legacy Labs in Mexico is the only fully licensed lab in North America making injectable cellular nagalase inhibiting factor, CNIF. CNIF will be administered in the Chipsa Combination 3-week program. Injections will be given at no additional cost to the patient during their stay. Due to the power of this treatment, patients looking to do CNIF outside combination therapy must be on a clinical trial program and are not available to the general public. All versions can be recommended, but have much less macrophage activation and nagalase inhibition, which is why Chipsa recommends injectable treatment. Combination therapy in 2021 at Chipsa may include injectable CNIF, Coley's toxins, cryoablation therapy, IV, oral and intratumoral apitone, high-dose IV vitamin C, autologous CAR T-cell therapy, autologous dendritic cell therapy with or without specific tumor antigens, autologous natural killer cell therapy, personalized tumor lysate vaccine made from the patient's own tumor, and other cutting-edge therapies. Call now if you or someone you love would like to learn more and speak to one of our doctors for free.